Hello everyone. Children, this is the B part of structure and germination of seeds. And children, in the previous video, you have seen or you have studied the structure of seed. As you know, children, the typical seed is differentiated into three parts, that is seed coat, embryo, and the stored food. And you have seen the generalized structure of a seed. Today, we will study about the structure of bean seed, that this bean seed is a dicotyledonous X-albuminous seed. Dicotyledonous means it has two cotyledons, and X-albuminous means that in this seed the food is stored in the cotyledons, not in the endosperm. So this seed is without endosperm. Reserve food is present in the cotyledons. Bean seeds are kidney shaped, being convex on one side and concave on the other side. Now see here, children. These are some of the bean seeds. You can see here they are kidney shaped. See this here. The shape of this bean seed is kidney shaped. Now, being convex on one side and concave on the other. The following external features are seen in the bean seed. Seed coat. Bean seed is surrounded by two seed coats that develop from the two integuments of the ovule. Now see here, children, in this bean seed you can see this brownish covering on this seed. This brown color cover is the seed coat, and this seed coat is developed from the integuments. As we have seen in the structure of ovule, that the ovule is surrounded by a two-layered integuments, and these two integuments form the seed coat. These two coats are testa. It is the outer, smooth, thick and colored protective layer. It protects the seed from fungi, bacteria and insects. See children, the outer integument forms the testa, and this testa is outer layer. It is smooth, it is thick. See children, you can see here it is smooth. This one, it is smooth and it is thick also, and it is colored also. Protective layer, it protects the main function of testa is to protect the seed from fungi, bacteria and insects. Tegmen, the second layer is the tegmen. It is thin, white membrane like inner seed coat. See here, this tegmen, it is thin, it is white, membrane like. It is very thin, just like a membrane, and it forms the inner seed coat. It is very firmly fused with the testa. It is often very difficult to separate it from the testa. So this tegmen is attached to the testa, and it is very difficult to separate it from the testa. It is so firmly fused with the testa that it is very difficult to separate it. So these testa and tegmen are both protective. Their main function is to protect the seed from infection of bacteria, fungi and insects. Helium. On the concave side of the seed, see here children, this one, this is the convex side and this is the concave side. This one is the concave side. Now you can see here On the concave side of the seed is present a scar called helium. See here, this one, you can see this white structure, this one. This, it is a scar. You can see here, this is scar, and this scar is known as a helium. It is the point from where the seed is attached to the pod with the help of a short stalk called the funicles. While we were discussing the structure of ovule, we have seen that this ovule is attached to the placenta with a stalk, and that stalk is known as the funicles. As you know, children, that after fertilization, the ovule changes into seed. So now the seed is attached to the pod with the help of a short stalk called as the funicles. On maturity, the funicles detaches and leaves behind this starch. So after the detachment of the stalk, this leaves a scar. You can see this white portion here. It leaves a scar, and this scar is known as the helium. Micropyle. Adjacent to the helium is present a very minute pore called the micropyle. As yesterday we were discussing about the generalized structure of a seed, there also I told you that adjacent or very lying very close or near to the helium is a micropyle, which is invisible. We cannot see that after soaking it in water, and if you will gently press that seed, you can see some water oozing out from that small pore and some air bubbles also. Then only we can see that micropyle. Absorption of water and exchange of gases takes place through the micropyle during germination of seed. So the main function of micropyle is to absorb water. When we soak these seeds in water, the water enters the seed through this micropyle, and after absorbing the water, the seed swells up. And after the swelling of the seed, when the seed swells up, then only we can remove the seed coat, and you can see, you can cut open that seed. So the main function of the micropyle is absorption of water and exchange of respiratory gases takes place through this micropyle during the germination of the seed. Next is RAF. It is a ridge-like structure present around the median groove. As you have seen here, this is the median groove, and here this is the median groove, we can see. It represents the portion of the stalk funiculus which remains fused with the testa. So this, it represents the portion of the stalk which remains fused. That means this portion remains fused with the testa. To study the internal structure of a bean seed, it is soaked in water for a few hours. So children, I told you that when we soak these seeds in water, the seeds absorb water through the micropyle and they swells up. The soaking makes the seed coat soft and easy to remove. On removing the seed coat, we find the embryo. Now, when you will soak the seeds in water, they will absorb water, they will swell, and we can easily remove the seed coat. And then you can open the two cotyledons and you can see inside it a embryo. Embryo. The embryo consists of the following parts. Cotyledons. The bulk of embryo consists of two fleshy structures called the cotyledons. When you will open that seed after soaking in water and removing the seed coat. You can open these seeds like this, and inside that you can see these are the two cotyledons, and this is the embryo. This is the embryo, and this one is the embryo axis. This is the embryo axis. It is differentiated into mesocotyle, a part of the embryo axis where cotyledons are attached laterally. Now see here, this is the embryo axis, and this one, these two cotyledons are attached to the embryo axis. So the part of the embryo axis where these two cotyledons are attached here, that is known as the mesocotyle. Apicotyle portion of the embryo axis between the mesocotyle and plumule. So see here children, this is the plumule and this is the mesocotyle. So this portion is known as the apicotyle. Plumule, the feathery or leafy end of the embryo axis that later grows into the shoot system. So children, this is the plumule, and in the later stages of its germination, it grows as a shoot system. So this is the plumule. After germination, in the later stages, it forms the shoot system. Hypocotyle, the portion of the embryo axis between the mesocotyle and the radicle. See children, this is the radicle, and the portion between the mesocotyle and the radicle is known as hypocotyle. Radicle, the pointed lower end of the embryo axis that later grows into root system of the plant. So this pointed end of the embryo axis forms the radicle, and in the later stages of germination, this radicle forms the root system. So this is all about the structure of a bean seed, which is a dicotyledonous X-albuminous seeds. So here we end this session. Bye.